Hello everyone, my name is Ava Laszlo, and today I will be presenting mussel assemblages in 4 reservoir lakes on the Tittabawassee River, Lake Huron drainage, Michigan, USA, after 2020 dam failures. The Tittabawassee River watershed is approximately 3,800 square kilometers and located in central Michigan. The Tittabawassee River is a tributary of the Saginaw River, which flows into Saginaw Bay and Lake Huron. Within the watershed, there exists a series of 4 reservoir lakes in Gladwin County and Midland County. These reservoirs were created in 1925 after dams were built 1923 through 1925 for the purpose of hydroelectric power. The names of these lakes are Secord Lake, Smallwood Lake, Wixom Lake, and Sanford Lake. Between May 17th through May 19th, 2020, record rainfall occurred in Midland and Gladwin counties. This record rainfall, combined with pre-existing dam infrastructure issues, led to failure of the Edenville Dam on Wixom Lake and failure of the Sanford Dam on Sanford Lake. Preliminary damages are estimated at over $250 million across the 4 lakes region. Almost 7,000 waterfront properties were affected, in addition to excessive damage caused to the city of Midland, the village of Sanford, and Edenville Township. The pre-disaster water surface of Wixom Lake is approximately 8 kilometers squared, and the pre-disaster water surface of Sanford Lake was approximately 6 kilometers squared. The post-disaster water surface of Wixom Lake is approximately 2.7 kilometers squared, and Sanford Lake approximately 2 kilometers squared. This is a surface area loss of approximately 70%, and the light blue color on the map that is overlaid on top of the dark blue color is representative of the current post-disaster water surface. After the flooding occurred, Secord Lake and Smallwood Lake were drawn down 2.5 meters shortly after the disaster to inspect and repair the dams after damage. Secord Lake and Smallwood Lakes were not completely drained, but they do remain drawn down. In June 2020, after the May disaster, the Michigan DNR called upon CME researchers to come out to the lakes and perform mussel rescue operations, but the overall scene was grim. The 4 Lakes Task Force is a nonprofit organization with members that include lake residents and industry experts. The 4 Lakes Task Force hired CMU to perform freshwater mussel surveys in Smallwood and Secord Lakes in 2021 and in Wixom and Sanford Lakes in 2022 through 2023. The Tittabawassee River watershed is lacking in mussel survey data, with the exception of a 1984 study by Hay and Drawdown, the freshwater mussels of the upper Tittabawassee drainage. The sites visited in this 1984 study are represented by the black dots on this map. You can see, this study was tributary focused and lacking lake data. Our study goals were to perform full lake surveys for unionids, determine the presence and absence of endangered or threatened unionid species, and determine threats to unionids, including those of invasive species. For Secord and Smallwood Lakes 2021, we used ArcGIS digital mapping software in order to identify random sites located within the lakes that were at least 50 meters apart from one another. We also selected targeted sites, including those upstream and downstream of the dam action area, historical sites, and tributary sites. Once a site was selected, we would drive to the access point or boat launch site. Once we arrived on site, our first and foremost concern was whether a site was safe and accessible. If not, we would take photos, record as much information as possible, and report the site as unsurveyed. We would then select the next site. If a site was safe and accessible, we would collect and record both abiotic parameter data and biotic data. We performed surveys for live unionid mussels, and we also collected unionid shells. We also looked for the presence of invasive species, including quagga mussel, zebra mussel, Chinese mystery snail, and Asian clam. Before I get into the results, I wanted to talk a little bit about challenges. Our number one challenge was access, and in each of the two lakes, Secord Lake and Smallwood Lake, there was one boat launch that we utilized with our 14-foot John boat. As you can see in these images, the boat launches were pretty washed out and made difficult to get the boat into the water. So once we arrived at the boat launch, we had to physically pick up the boat and motor and place it into the water instead of as you would normally just back up a boat and trailer. The bottom image, you can see two docks that are visibly out of the water, and the red arrow is pointing at an area where we would bring the boat down into the water before launching. Due to the low water levels of Secord and Smallwood lakes, navigation was an issue due to exposed stumps. We also encountered thick macrophytes and algae and muck, in addition to Dracena polymorpha zebra mussel substrate. For our results, we surveyed 59 sites. These sites included 2 historic sites, lake sites, peripheral sites, and downstream sites. For Secord Lake, our live species, we only found one species, and that was Pyganodon grandis. We found a total of 305 individuals. For Secord Lake shell species, we found a total of 7 shell species, 6 that were shell only. For Smallwood Lake, we found 6 live species and 143 total individuals: Pyganodon grandis, Lasmagona castata, Utterbachia imbecilis, Lampsillus silicoidea, Lampsillus cardium, and Venusta ellipsiformis. For Smallwood Lake shells, we found a total of 10 different species, 6 that were shell only. We produced a species accumulation curve in order to determine the number of sites, the number of additional sites that we would have had to have visited in order to potentially detect one additional species. We discovered that we would have had to have sampled 20 more sites and spent 32 more person hours to potentially detect one additional species and go from 6 to 7 live species. We performed the same analysis with the number of individuals detected instead of number of sites visited, and found that we would have needed to find 410 more individuals to potentially detect one additional species. For our Secord and Smallwood summary, we found a total of 13 species represented by live or shell. 2 species, Lasmagona castata and Utterbachia imbecilis, were found live only, and they are also new records for the Michigan Natural Features Inventory for Gladwin County. 7 species were found shell only. We found no significant differences in Wentworth scale substrate variables when comparing mussel present versus mussel absent sites for both lakes. We found that sand, silt, and muck combined accounted for 75% to 85% of bottom habitat. The image here is representative lake bottom substrate in Smallwood Lake, but similar substrate was also encountered in Secord Lake. We looked at Pyganodon grandis size distributions between the 2 lakes. On the y-axis, we have percent of total individuals found, and on the x-axis, we have size class in millimeters. For Secord Lake, it is an approximately normally distributed curve. For Smallwood Lake, it is slightly skewed to the right. However, in Secord Lake, we found 0 or few individuals of the smaller size classes, while Smallwood Lake, we found 0 individuals of the larger size classes that were present in Secord Lake. We looked at the presence and absence of zebra mussels in both lakes at the sites and found that 90% of sites in both lakes had live Dracena polymorpha zebra mussels. However, in Secord Lake at the sites were denser with zebra mussels than in Smallwood Lake. We also looked at biofouling of unionids by zebra mussels. In Secord Lake, 100% of sites that had live unionid mussels, unionids were fouled by at least 1 Dracena polymorpha. In Smallwood Lake, 81% of sites that had live unionid mussels, unionids were fouled by at least 1 Dracena polymorpha. When it came to the density of unionid fouling by Dracena polymorpha, Secord Lake unionid specimens were covered in a denser aggregation of zebra mussels than in Smallwood Lake. As far as historic sites go, Hay and Drawdown in 1984 at the northernmost historic site found 1 shell species, whereas when we went back to that same site in 2021, we found no shell and no live unionids. For the second historic site, Hay and Drawdown found 7 live species, and when CMU went back in 2021 to the same site, we located no shell and no live unionids. We found clear evidence of habitat alteration after a drawdown of the lakes, for example, successional plants such as willows and grasses. We found patchy distribution of common lentic or lake-adapted species with low species richness. The dominant unionid in both Secord and Smallwood lakes was Pyganodon grandis, or giant floater. We noted often heavy biofouling by zebra mussel on unionids and hard surfaces. Lastly, we noticed a large population of invasive Chinese mystery snail at several sites, especially in Secord Lake. As far as the future goes, in October 2021, CME researchers performed a reconnaissance trip to both Wixom and Sanford Lakes with canoes in order to better understand the areas that we will be surveying the following 2 summers. In December 2021, CMU will deliver a report with our findings from this year's research to the 4 Lakes Task Force. I would like to thank our funding source, the 4 Lakes Task Force. I would like to acknowledge and thank all of the field and lab help this season. And lastly, I would like to acknowledge all of the agencies that were involved with the logistics and feedback for this research project. If you have any questions or comments, please feel free to contact myself, Ava Laszlo, Dr. Dalen Woolnough, and Dr. Dave Zanatta. Thank you.